Hello, hi, it's DrBiology here, and this is a video on infection and response. Thank you. Right, okay. Right, let's get started then. So first of all, we're going to look at pathogens. Okay, so we're going to talk a bit about pathogens. Can anyone give me a definition, please, of what a pathogen is? What is a pathogen? Yeah.
A microbe that causes disease.
Yep, a microbe that causes disease. Well done. So we're going to talk about microbes that cause disease. Because not all pathogens cause us disease, but we're interested in the ones that do. Okay, and there are 4 types. We're going to talk about each of them individually. So here, first of all, we're going to go bacteria. And bacteria, there are 2 types of diseases you need to know about bacteria, and they are gonorrhea and salmonella. Okay, so gonorrhea, the main things related to that is a yellow-green discharge. It's an STI. Sexually transmitted. Salmonella, okay, so salmonella is basically you get nausea, vomiting, abdominal cramps, fever, okay, you get it from eating infected food. So you can get fever, diarrhea, like gonorrhea. Okay, and yeah, they release toxins. So they're right. Okay, let's go down here. We'll talk about viruses. We're saying that they live inside host cells. Okay, and there are 2. So we need to know about the 2 types of viral disease. First one is HIV, or human immunodeficiency virus. And it's transmitted by bodily fluids. Blood, things like that. Things like blood. Also from sexual contact. The problem with HIV is it attacks the immune system. It can lead to AIDS, which is acquired immunodeficiency disease, or syndrome, sorry, it's not a disease. And it can eventually cause death. All of these can cause death. Going to be a bit confusing otherwise. Second type of thing is measles. So you get fever, you get red rash. It's spread by droplets in the air, so droplets in air. And the other one is TMV, which is tobacco mosaic virus. So that's in plant. It's a plant. So obviously plants get diseases as well. Put it over there, so you get mosaic leaf. So yellow patches. And you have to destroy the infected plants. Okay, so far we've got bacteria, viruses, where the next one we're going to go for is, and I'm going to do it here, I'm going to go across here, and that is protists. And there's only one type of protist you need to know about, and that's something called Plasmodium that causes malaria. So you get malaria. It's spread by mosquitoes. So again, you can get fever, vomiting, muscular pain, and death as well. Pain, death. So you can stop malaria. So how do you stop it? Well, you stop by killing the vectors. So you can use, well, you could use nets to stop them biting you. You could use pesticides, insecticides to kill the mosquitoes. You could remove water. Okay, and then the last one, I'm going to stick it around here, and that is the fungi. Okay, now fungi, you can get athlete's foot, which is not very nice, but in plants, okay, so there's another plant disease you need to know about, is rose black spot. Rose black spot. And as the name suggests, you get leaves that have black spots on them. Okay, and so obviously that's going to reduce photosynthesis. How they spread? It's spread by the wind and by droplets. Okay, and you can use a fungicide to treat it. So very briefly, those are basically all the pathogens. So the 4 pathogens you need to know about, which are bacteria, viruses, protists, and fungi. And those are all the diseases you need to know about for the exam. Right, okay, human defense system next. So let's go to the defense. So we're going to go up here, and I'm going to go for defense, microbes. So let's look at the non-specific first of all. We things like skin, hair, so in your nozzle, your nose, your trachea, your windpipe. Mucus, again, same places. Stomach acid to kill pathogens, and also your blood clots as well. So when you cut yourself, blood clots and scabs. All right, so that's non-specific. Let's talk about the specific. So I'm going to do specific. So we're talking about the good old WBCs, white blood cells. So they specifically produce 3 things. Okay, so the first thing is they produce antitoxins to neutralize any toxins from bacteria. They produce antibodies. Now we'll talk about antibodies a bit more, but they're specific to a particular disease. And finally, they engulf, or better word, phagocytosis. Okay, so basically they're engulfing. So So they engulf bacteria. Okay, they digest them. And they also display the antigens. So obviously with WBCs as well, you get natural immunity. So this is called natural immunity. Good, right, okay, I'm going to go on to vaccinations now. So vaccination is basically you need to know that they are dead, weakened, inactive, we even say pathogens or parts of pathogens. Let's say pathogens stroke antigens, because they have antigens on them. Yeah. What does it do? Well, the vaccination, it means that the WBCs produce antibodies, specific, sorry, let's use that word, specific antibodies. Okay, they remain in the bloodstream or remain in the body as memory cells. So when you're infected, so infection means the body reacts faster, body produces antibodies, let's call them ABs, faster and produces faster and lots are released. Okay. We are going to now look at antibiotics. I'm going to do antibiotics really quickly. Right, so what are antibiotics? Well, you can have things like penicillin. Okay, so penicillin tends to be fungal molds, could be other things, but fungal molds. Okay, this is the big one. They only kill bacteria. Only kill bacteria. Okay, they can't kill viruses because the viruses are inside the cells. Only kill bacteria. Okay, however, there are some disadvantages of antibiotics, as we've said. So this is the DISADS here. So can't kill viruses. Some bacteria are resistant, so you get resistant bacteria. They're caused by random mutations, remember. Okay, so let's do about drug development. So new drugs. So new drugs, very, very quickly, you can have, well, where do drugs come from? Well, you can have things like digitalis, which is from the foxglove. You can have aspirin, which is from willow trees. Okay, so willow. You can have, what else can you have? Penicillin. So they all kind of originate from plants, but the thing is, they all going to, well, these days, so modern ones are synthesized. Modern are synthesized in a laboratory. Do you know what I'm going to do? I'm going to rub that out and go over there. I know. I know. It's not great, is it? Sorry. Different stages. So I'm going to go here. So the stages of drug development. you need to know. Number one, you synthesize in a laboratory. You test thousands of compounds. You can do computer modeling. You might use some tissue. Stage 2, we go to animal testing. So we're testing for two things: toxicity. In other words, is it toxic? But also, it's whether it works. Does it work? It's mainly that toxicity with animals. So that's known as preclinical. Then we go stage 3, which is clinical. These stages can be different in different ways. Well, so this is with healthy humans. And they're basically given a low dose of the drug, low dose to test for side effects, for safety side effects. Then we go to the next stage, stage 4. And these are large clinical trials, large trials. So they're looking for optimum dose and what we call efficacy. Efficacy just means whether it works or not. Okay, and then we go stage 5, in this case, which is going to be registration. So it needs to go through lots and lots of checks with the appropriate bodies. So other scientists check it. So scientists check the results. And then you can sell it. So all that is going to take, it takes 10 to 15 years for most common drugs to do all that. What have I missed out there? Oh, I've missed some really important things, like double-blind trial. So I'm just going to mention double-blind trial. So these are keywords. So double-blind trial. So this is where neither doctor nor patient know who's had the drug. Okay, so that avoids bias. Okay, and then another important word is placebo. Placebo. So placebo is like a dummy drug. Okay, it looks and tastes the same. So you don't know whether it looks and tastes the same. So you don't know whether you've had the real drug or the placebo. Okay. And again, that avoids bias. Good. Right, so if you do combined science, that is the end of infection and response. So at this point in the video, you can stop and review. Thank you for listening. I'll see you soon. And action. Hello, DrBiology here, and this is the second part of this video and is for GCSE biology students. So this is the extra work you need to know for infection and response. So let's go through it together. So first of all, you will need to know a bit about monoclonal antibodies. So that's the first thing we'll look at. Mono meaning one, clonal meaning it's genetically the same. So just one type of antibody. So monoclonal antibodies have lots of uses in science, and we're going to talk about how they're made. So first of all, how are they produced? Okay, in your GCSE, you don't need to know very much about how they're produced, but I think it's useful that you do know how they're produced, so I'm going to tell you. So they vaccinate a mouse, okay? They then remove the white blood cells from the mouse. And unfortunately, yeah, that's the end of the mouse. And then what they do is they fuse, so they have the white blood cell. So here's the white blood cell, WBC. And they have a cancer cell, a tumor cell. So what they do is that they fuse the white blood cells, the lymphocytes, to the cancer cell. And that makes what we call a hybridoma. Hybridoma. Okay, now this hybridoma is useful because then you can produce the specific type of antibody, produce the specific type of antibody. Because it's a clone. So it's a cancer cell, so it divides very quickly. So it can divide quickly and produce large amounts. So I haven't draw the antibodies. In this case, this is what the antibody looks like. So the hybridomes produce loads and loads of these antibodies. Okay, so what are they going to be used for? So we need to know a bit about uses, but I'm going to give you a top tip first of all. Okay, so this is a Dr. B top tip. I'm going to put it here. And the top tip is this, is use the information given in the question. They like you to evaluate things as well. So they might want you to evaluate information. So just a reminder, to evaluate means to look at the pros and the cons. So pros and cons of something, okay? But it doesn't hurt us not to go through it very quickly. So what are they going to be used for? Well, they'll be used for research. So they can maybe target cancerous cells. So it's used in cancer treatments. So they can get the antibodies to the cancer cells, for example. So that's the second one, is for curing disease, so treating disease. So you can attract immune cells to an area, to a site of infection with them. So if you release lots of them in one area, you get loads of all your immune cells going to the same place. As I said, you could use it for cancer treatments. So delivering a drug using the antibody to the cancer cells. It's used in pregnancy tests, okay? And in fact, let's talk about COVID tests as well. So it's used for lateral flow as well. They might talk about, I don't think they will though. If you're watching this in 2024-25, they might ask you about lateral flow tests. There you go. So pregnancy, lateral flow tests. So the monoclonal antibody is on the test strip, okay? And basically the antigens attach to it, to the antibodies, and a colored dye is released by an enzyme. So that's where you get the characteristic two. So in a lateral flow, you've got the control and the actual test strip, and it changes color. So that means it's worked. That means you've got it. It's a double line. As some of you will well know. And it also can be used for disease diagnosis as well. So disease diagnosis, so thrombosis, stuff like that. So things like heart disease, thrombosis, which is blood clotting. Okay. That's where they probably give you lots of extra information. Cool. Right, okay, very, very quickly. Last one is plant defenses. Okay, so defenses from disease. We talked about humans. Let's talk about the plants. I haven't got time to draw plants, so get on with it. Stay with me, please. I've got 5 minutes of this. Right, okay, so plant defenses. We'll talk about them. So you get 3 types. Mechanical, so that's kind of spines, spikes. So physical things that are in the way, okay? Physical, so you got waxy cuticles, produce, so very thick leaves. They got thick cell walls. They lose leaves, so deciduous trees lose leaves. Okay, so it's a physical thing. Chemical, poisons, sap. So saps that are sticky. Remember that video? Antibacterial chemicals, stop infections. There we go. Right, another top tip, they'll give you plenty of information in the exam about that. They'll probably show you a plant you've never seen before. Look at it very carefully. Look for the clues in the question to help you. Diseases, very quickly. Again, you know lots about plant diseases already because you've done it before, and I'm not going to talk about it again. So tobacco mosaic virus, rose black spot, the new one in this section is aphids. So aphids are green fly, okay? So aphids, problem with aphids are they spread viruses, okay? They also damage growth, plant growth. Okay. And finally, my goodness me, this has been the longest lesson ever. Ion deficiencies. You're doing very well, class. They are good, aren't they? Yeah. Right. I'm not kind, but there you go. So nitrate, I am actually. Nitrates. Okay, so nitrates important for protein synthesis. So proteins, makes proteins. A deficiency means they can't make proteins. So the symptoms, main symptom I'm going to talk about is stunted growth. Okay, so it reduces growth. And then the other one you need to know about is magnesium. And that makes chlorophyll. So obviously you're going to get yellowing of leaves. And you can treat them with fertilizers. Fertilizers. Oops. Fertilizers. Okay, so that was a whistle-stop tour through infection and response, both for combined science students and GCSE biology. Thank you for listening. I like you lot. You're very good. Well done.